I want to thank you all for coming and the opportunity to talk today. So it kind of blows me away to be going after Armin because their work is so fantastic. And he said he's not going to talk about the old stuff. That gives the old stuff field wide open for me. So I'm going to be talking really about what we're doing sort of at the ground level. And I'll give you a little background about where we are as a lab. So I've been responsible for generating the extractable leachable for the new products and also for backfilling some of the legacy products. We had a lab in Belgium, not far from here, that would be doing the extractions for customers and the validations for customers. Unfortunately, that lab was shut down. Li Jun, are you here? Li Jun joined Nelson from that lab, and so we are currently doing some of that work here, but the old stuff still continues to be on our plate. And so I was talking to my colleague Noel about what we present here, because everyone's so familiar with the products and the extractable leachable process, and said, I don't know, is anyone going to be interested in this? And so we were looking at the agenda, and I said, at least Dennis Jenke's not going to be here. And she said, you didn't look at the second page of the agenda. So I am going to be teaching you what you told us about in the book already. So here, let me give you a little bit of background around where I'm coming from in terms of looking at extractable leachables. So my first job out of graduate school was measuring paint film thickness on cars. And you didn't think you were going to hear about paint today, did you? Because it's a very complicated process. You spray it on. The variables are very large in terms of producing a quality finish. There's temperature, there's humidity, there's particle size, and if Ed is not there one day, everything can fall apart. Very complicated. And so the challenge of being able to control something, you have to be able to measure it first. And that measurement is key to all sorts of things that flow after that. Now, after that, I've worked for Pfizer in PAT, measuring applications for granulation and particle size and crystal forms, polymorphs, any number of things. But the same thing applied. Oftentimes in these, especially in small molecule, in the mixing and solid material, the process is variable in ways that are sometimes unpredictable. And so being able to measure that gave you power into what you ended up at the end. And so this capability of measurement and understanding that you cannot control something you do not measure is something that's informed my approach to all sorts of things. After that, I've been at Pfizer or at 3M for the past 15 years or so, working on first in corporate analytical, where I worked on root cause analysis on all sorts of manufacturing failures. 3M. Did I say that 3M has manufacturing failures? I didn't say it. We have unresolved issues, occasionally unresolved issues. But it sensitized me to understand that there are all sorts of manufacturing challenges that if you don't understand them, if you don't measure them, you can meet all the specifications that are set for a specific product and still not achieve the quality aspects that are important. So ultimately, it's about measuring those. And so I'll cut to the chase on this. We don't measure extractable leachables all the time. It's not a specification, even though we sometimes suggest that it's something that will come out every time. So I am going to start. by briefly going over the product portfolio that 3M has. Now, it's a very different slate of materials than some of our other speakers have talked, because we're basically only filters. We don't build constructions out of these. We don't have bags. And so we are only making filters which the customer then can use in whatever place they want. But I think this will be an instructive suite of materials that we can talk about the approaches to extractable leaches in any individual one of these. So we have sterilizing grade, which I'm going to talk about a little bit. We have a depth filter, and so that will be, the well-known problems with depth filters, but I'll highlight that a little bit. And in the third case, on the bottom is our suite of all synthetic new materials. And I'm going to point out this one, and I'm not no advertising. So this is replacing centrifuging for cells. I just love this SEM picture because it shows the cells on the fibers. So as an analytical chemist, I just love showing SEM images. All right, so as we've spoken about in a variety of talks up to this, really the importance of understanding extractable leachables is really where the component is in the process. And I have worked through with a number of customers, and it always seems that the customers further up are always concerned about performance, whereas the ones at the bottom are obviously much more concerned about the safety concerns. And so that has been reiterated a number of times, and also the opportunities for the extractables to be removed during this process. So I wanted to give a brief overview of how we do extractables at 3M. Not that it's earth-shattering, but we have always followed USP 665 as closely as possible. There were early days when we were doing the BPUG solvents. There's no reason to relitigate that. This solvent volume to filtration ratio, typically 3-to-1 is about as good as you can get, even though the recommendation is 1-to-1. If you want to chop it up and do little pieces, you could probably get 1-to-1. I have been dedicated and committed to doing only full filter as much as possible. I am quite sensitive to the possibility of flow making quite a difference in the ultimate extractions that you get. And if you do that on an actual filter, you will be getting much more representative data than if you don't. And we continue to do 7 days. Now I know this is not required by the regulations, but we found that it gives you a lot of very interesting data. First of all, customers will say, the conventional wisdom is that nobody's going to use a filter for longer than 24 hours. Oftentimes, a customer will use a filter for longer than 24 hours. Sometimes they will preconditioning flush it and then let it sit for a day. Maybe they'll have a process interruption that'll go on longer than that. So it's always better to have something that encompasses challenges that the customer may have. The other advantage of having 7 days is that it really does highlight that mechanism for generation of the extractable. So sometimes it'll go down because the filter is reabsorbing it somehow. Sometimes it'll remain flat. Sometimes it'll have a curve. Sometimes it's being generated by the material. So this is especially true in some of the ligands I'll talk about later. But having that extra data point, 2 data points is nothing. You don't understand anything having 2 data points. And so classically, as an analytical chemist, the mantra I have is 1 data point is 0 data points. 2 data points is also 0 data points. And 3 data points is now 1 data point. So you can understand more about it by having just that extra day. All right, so I'm going to take just a brief second to talk about validation for sterilizing grade filters. Now, Peter Cornelis is going to be talking about this tomorrow in much greater detail. And this is something that we have transferred to Nelson as a standard testing protocol. And the reason I bring this up, first of all, conceptually, it's really straightforward. So you've got holes, you have bacteria that get caught in the holes, and conceptually, this is what's happening. But in reality, the filter media is much more complicated than this. So it always irks me a bit when you say you have 0.2 micron media. It's really a nominal amount. And so that tells you how much it's going to filter out. It has nothing to do with, or it doesn't have much to do with pores. These are not isoporous membranes. These are complicated structures. And so we test bacterial challenge by seeing what will go through. We also have integrity tests where you look at where the bubble is. And so every single sterilizing grade filter needs to be validated with the filtered fluid that the customer is going to use. Must be validated to confirm that the bacteria are filtered out for that fluid. And so the failure, pass-fail criteria, is one bacteria, one colony-forming unit. So there is no range of colors in this analysis. It's only pass or fail. Oops, sorry about that. And so I think this is the difference between validating for a specific test like this, where you will fail, excuse me, versus the range of things where the analysis is the extractable leachable analysis is something we provide that the customer can accept and apply in specific areas. Now, the one thing that I believe Mona talked about earlier, and the thing that irks me significantly, is that I am always talking about people within my own organization. They ask me, well, does the filter pass BPOG? Does it pass USB 665? And there is no pass. There is only providing that data to the customer. And I continue to try to teach people that there is no pass, but I have only been partially successful. So the extractable leachable portion of sterilizing grade filters, I have a quote from Armin here. Because this is essentially a solved problem. We extract the material. The foundation of the generation of these membranes is quite robust. The process is robust, as opposed to many of the other processes that, painting cars, wet granulation. Some of these are more complicated, and the process itself is not very well controlled. But sterilizing grade filters, this is a done deal. So thank you for the quote. This was someday I'll give a talk that only features data that you've done. So that could be quite easy. So the next product I'm going to talk about is depth filters, and I'm sure those of you who have looked at depth filters have all understand their complexity and their challenges. And there's always been a push towards making more all synthetic materials. But I would love to tell you exactly how much we still sell in depth filters and how much that is continuing to grow. So you can talk about all synthetic materials, but depth filters are here to stay. And they've been spec'd in as so many applications that it's going to be a long time before we get rid of these. So for those of you who might not be aware, depth filters are essentially dirt, sticks, and glue. It's complicated, expensive dirt, sticks, and glue. So cellulose, all right, it's low alpha cellulose and epichlorohydrin resin and diatomaceous earth. It can be acid washed, or there's a variety of different ways. But the resin is extraordinarily complicated. And this is just data from an original scouting where the number of resin fragments is overwhelming. and it's also unpredictable. So we can extract one grade of material, and there's low levels of extractables, and it's very difficult to provide something that's going to be the same. So with a sterilizing grade filter, you're going to get caprolactam, you're going to get butyrolactam, you're going to get a few other things, and it's all very straightforward. And you know exactly where they're coming from. I know exactly where these are coming from, but am I going to guarantee that you're going to get the same resin fragments with another material, with another filter that's processed similarly? And this is cooked up in a giant vat, like spaghetti. And so it's with the natural materials that are always varying. I'm going to go back to the statement I started with. These are not specifications, and so we're not controlling these because we don't test for them every single time. And so it's interesting. I'm having a customer right now who is complaining that there are too many beta glucans in our depth filter. You know, beta glucans come from cellulose. And I tell the customer, you know, we don't specify that. And he says, but there's more. And I said, we don't specify that. And so this back and forth, just because something changed, I don't know what changed. Maybe it was something about the wood that was being harvested at that specific time. But he's seeing this and saying that changed. I'm concerned. Why? Can you tell me why that changed and how? And so I say no, because we don't specify it, and that's ultimately the answer. But it's hard, and the question I have, which is a follow-on, we are constantly in the process of making modifications. We are developing new manufacturing sites, and we do equivalent studies with extractable leachables because everybody wants extractable leachables now. You guys have done a great job getting the customers to want the extractable leachable data, and so we will make this change, and the customer will say, all right, let's compare that to the extractable leachable data that you had before. And I don't know how to specify pass or fail based on these changing numbers. So again, with sterilizing grade filters, it's easy. It's going to be plus or minus 5% because the manufacturing process is fully in control. But when we have something like this, where there's variation all over, they start looking at this and saying, this is not the same. I said, this is not a specification. This is a guidance for you. But that's a hard message to tell people who have qualified things, who are in quality, that I'm not saying anything bad about quality, it's very important, but they're not used to having something that's provided as a guidance rather than an analytical number. And so I add this one. This is mostly so you feel sympathy for me. So before we had extractable leachables, 3M made all sorts of products. And so we have 8 different media families. We've got all of these are different varieties of depth filters with different flavors. Some have different resins. Some of them have different cellulose. Some of them have different diatomaceous earth. Some of them are acid wash. Some of them aren't. And so we can have different sanitization methods. And then each one of these has at least 6 different media grades. And then we make products that combine these media grades together. So we have a thin one and a thick one. And so imagine going to a new manufacturing site and having to do extractable leachables on every single goddamn one of these. It's suddenly a huge, huge amount of work. And so at this point, what we've been doing is one representative grade. And then telling the customers, well, this is going to be pretty close. This is a guidance on what kind of material you should be looking for in your product. But it ends up being a question of risk rather than an analytical measurement. So we are not saying that this is the number you're going to get. This is the kind of material you should be looking for when you are qualifying your own product. But I look at this and hope that, as we become more mature in our 665 approach, that customers will be able to see this and say, I don't need every single one of these grades, but I'm afraid that they might. So I hope you're feeling sympathy for me, because this is a tremendous potential amount of work for us to. And so the third sort of category. So back. This is something I would call not intractable, but maybe from an analytical standpoint. This is so difficult to come up with something that's completely answerable to something that's more interesting. So this is our all synthetic material that has ligands on it. I'm not going to tell you exactly what kind of ligands, but interesting chemistries. So just to start off, one thing I've noticed, and these are interesting chemistries. I'm responsible for distributing these extractable leachable reports to our customers, and I will send out all sorts of reports on our depth filters and other ones where I expect people to just accept those. I'll send out these reports, and the customer never comes back to me with questions. And there are these very novel molecules, some of which we would only talk about under CDA. I describe them in sort of generalized terms in the extractable report. But I've been waiting for someone to come back to me and say, I'd like to know more about this. Can we discuss this under CDA? And no one ever comes back to me. So I'm not lonely. I don't feel, but I'd like my work to be validated something. So I know that a lot of you are working with folks and doing the tox assessments, and I know there's lots of people out there doing great work. But I'm thinking that this maybe speaks to the heterogeneity of the response by many of the drug manufacturers out there. And so this is often concerning to me, is that this ends up being a checkbox rather than a real opportunity. And hopefully the new 665 guidance will do that for us. But so the challenge here is that, let's say with the first one, the sterilizing grade filters, the manufacturing process is so understood that we don't even need to worry about it. Depth filters, the manufacturing process is variable enough that we're not going to worry about that so much either. Not out of control, but the analytical is hard. And so this is a little bit in between, because these are novel materials with novel manufacturing processes, and maybe we don't, I would never argue that manufacturing processes are unknowable, but sometimes the variables are not yet well understood, and you honestly just can't test all the variables. You can test some of the obvious ones, but moving forward with that, you couldn't have any manufacturing process where that you have start for the very first time where you understand everything. I wish that was true. But for a novel material like this, we have set up a variety of different tests that allow us to feel very confident going forward. And so obviously we have viral clearance, which is one of the primary claims. We have binding capacity to make sure that the performance is okay. But I want to draw your attention specifically to the green one. So ultimately, extractable leachable is partly about performance, partly about patient safety. But the primary claim for all of these, performance is great, but patient safety is the claim. Is either it's going to be safe or it's not. And so for this material, the ligands are all nitrogen containing. So we have made a very distinct correlation between USP87 data and the amount of total nitrogen that's being given off to this product. So typically, if you were going to qualify a product, you would run 87 or 88 on production batch and then call it good. But to get us to that extra point where we feel confident of a process that we are, we feel pretty good about it, but to take us to that extra level where we are 100% confident that this meets all toxicity and all patient safety requirements straight out of the box, we essentially revalidate USP 87 every single time that we create a batch of the material. Which I think it's a powerful opportunity. So instead of just saying, regulatory requirements require us to do this, we have the extractables, and that's good enough. You can take it to that next level where maybe you can do extractables on every single product. Maybe not the same kind of thing. Maybe you're not going to run MSMS on every single batch. But you can maybe have surrogates or something that'll give you an opportunity to improve your confidence. So, just a few things. So, extractable problems are different and require different mindsets. And so, being able to work through that is important to understand. So, there is variability in manufacturing, and I love the data. Everyone is doing the tox assessments based on a small subset of filters or a small subset of materials. Did I mention it was not a specification? I think I did. So I asked this question a few years ago of Dr. Jenke, and what is the number that we're being held to? And if I remember the answer correctly, it was something that the manufacturing process should be in control sufficiently to, and if everything else being equal, the extractables should be consistent. But I'm going to say again, based on my experience in manufacturing, if we're not testing for the extractables, we can have some confidence they'll be the same, but that's not the specification. They could be floating over here, up and down, and that float ends up being larger the smaller the extractable is. So caprolactam is probably going to be not going to be changing very much, but something that's 10 parts per billion or 5 parts per billion, I don't even know if that's going to be present all the time. So that the confidence in that number, don't overestimate it. But I'm not recommending that we do release testing based on extractable leachables. I will not. I will find another job before we do that. I said that again. So this is, but I'm going to say it again because it's an important message to the customer. That would be you, drug manufacturers, that this is a guide for you to be able to do your own assessment. And so when I was getting this talk approved, this was the line that got me in trouble because regulatory has to approve it. And they said, what does this mean? Don't let regulatory get in the way of doing the right thing. And I said, oh, that sounds horrible. So Tammy Johnson, if you're watching this this video, it wasn't you. This is specifically in regards to being able to measure more than you need to. So just because you've met the regulatory requirement doesn't mean that there aren't other things that you should be doing. So customer requirements are oftentimes unpredictable. And so the what they expect in terms of extractable leachables can vary wildly. So sometimes you can write a letter that says, your process is fine. I think you should be good using the data that we have. And they'll say, that's great. And then other ones will say, I need you to test the extractables on this data specifically. And getting that aligned would help everybody And so how can we keep it from being a quality checkbox? I'm not sure if there is a way around that, because not that we need to have a way around that. I heard a talk from Dr. Jenke about, do we still need to measure polypropylene anymore? That's been a few years now. Do we need to still measure polypropylene? Whether we need to is a scientific question or not. It's about whether the customer is going to require it. So I've changed polypropylenes probably 5 times in the last 2 years, and every single one of those has a pretty extensive extractable leachable package because we've got customers demanding it. And so what the customer demands, the customer will get. And I also want to give kudos for not requiring USP 87 or 88 anymore, but that's got to be a industry-wide acceptance, because we can't have one company that says 87, another company that still requires 88. That means all the vendors need to do 88. So now, how do we maximize the impact while minimizing the cost? Now, this is how much cost does this really add to the final product? I'm not sure, but this is the horse that I like to ride in on, because there is no going back on regulation. It's always increasing, and putting the safety part of that together with manufacturing efficiency, and so they got a little bit smaller. So this is where I would like to be. That my own career would be able to say that I made things a little better or a little cheaper, and so unfortunately there's not a safety thing here. So I've given a variety of talks about drug pricing, and I would like to think that ultimately the work that we're doing is going to make these therapies more affordable and at the same time safer. So just to give you a brief, so this is about what monoclonal antibody therapy would cost. So this is actually my daughter, who's been on both monoclonal antibody therapies for large B-cell lymphoma and juvenile arthritis. So I have a vested interest in these products being both safe and effective. But also, you look at how much they cost. Now, how much, if we didn't have to do extractables leachable testing, that's not going to really affect these prices very much. But from an overall standpoint, getting the amount of testing we do right, not over testing, not under testing, but to get that amount right, that's where I want to be successful. So.